Good morning. It's January 14th, and this is your daily brief in science. Here's everything you need to know. A new groundbreaking test for lung cancer could change the way we approach treatment. A collaborative team from the Francis Crick Institute, UCL, UCLH, and Personalis has developed a test that detects circulating tumor DNA, or ctDNA, to predict outcomes in lung cancer patients. This development was supported by a Cancer Research UK-funded study. The Next Personal test has demonstrated high sensitivity, allowing for accurate detection of ctDNA and preventing misclassification of patients with low ctDNA levels. Research involving blood plasma samples from 171 individuals with early-stage lung cancer found that lower ctDNA levels before surgery were associated with reduced relapse rates and improved overall survival. Marianne Baker from Cancer Research UK stated that the Next Personal test represents significant progress towards personalized medicine, enabling tailored treatments based on individual disease progression. Charles Swanton, a senior author of the study, pointed out the challenges of treating lung cancer due to its high relapse rate, emphasizing the need to identify patients who might benefit from more aggressive treatment. Typically, treatment for early-stage lung cancer involves tumor removal through surgery, followed by chemotherapy or immunotherapy. The ongoing TRACERx-EVO study is currently recruiting participants to further investigate lung cancer biology and validate these findings related to ctDNA. The study's findings were published in the journal Nature Medicine, marking a step closer to personalized treatment approaches for lung cancer patients. James Black, a postdoctoral clinical fellow, noted that ctDNA presence in blood samples serves as a strong predictor of patient prognosis, potentially guiding treatment decisions. Future assessments will focus on ctDNA presence in post-surgical patients to evaluate its potential in predicting relapse risk and guiding additional therapies. Richard Chen from Personalis emphasized that the Next Personal platform aims for the early detection of residual or recurrent cancer, which could lead to timely interventions and reduce unnecessary treatments. Overall, this study suggests that sensitive ctDNA testing could significantly improve survival rates and quality of life by effectively guiding treatment decisions. Researchers have made a significant discovery regarding the Sydney funnel web spider, revealing that what was once thought to be a single species is actually 3 distinct species. These newly identified species include Atrax robustus, Atrax montanus, and Atrax christenseni. Atrax montanus is found in the Blue Mountains, while Atrax christenseni, the largest of the 3, is located near Newcastle, approximately 150 kilometers north of Sydney. This groundbreaking research was published on January 13, 2025, in BMC Ecology and Evolution, with contributions from scientists at Germany's Leibniz Institute, the Australian Museum, and Flinders University. The study employed anatomical and molecular DNA comparisons to differentiate between the funnel web spider populations. The investigation was initiated after unusually large male funnel web specimens were discovered in Newcastle, prompting the identification of the new species. Atrax christenseni was named in honor of spider enthusiast Kane Christensen, who played a crucial role in its identification and specimen collection. This finding holds significant implications for venom research and the development of antivenins, according to the Leibniz Institute for Biodiversity Research. Dr. Helen Smith confirmed that the antivenom developed in the 1980s continues to be effective for bites from all newly identified species. Since the launch of the antivenom program in 1981, Australia has not recorded any fatalities from funnel web spider bites, demonstrating the effectiveness of existing treatments. Professor Jeff Isbister noted that while bites from funnel web spiders are rare, they can be severe, emphasizing the importance of seeking immediate medical assistance if bitten. Additionally, the Australian Reptile Park is the only facility in Australia that extracts venom from male funnel web spiders, which are significantly more toxic than their female counterparts. Professor Chris Helgen from the Australian Museum commended the research for highlighting the importance of museum collections in modern scientific studies. Researchers at Chalmers University of Technology in Sweden and the University of Maryland have unveiled a groundbreaking quantum refrigerator that autonomously cools superconducting qubits to record-low temperatures. Operating at a remarkable 22 millikelvin, this refrigerator boosts the probability of qubits achieving their lowest energy state to 99.97%. This enhancement is vital for the efficiency of quantum computations, as quantum computers require extremely cold environments, near absolute zero, to function reliably. The compact design of this quantum refrigerator facilitates easier integration into existing quantum systems, making it beneficial for both research institutions and commercial enterprises focused on quantum applications. Notably, it operates by harnessing ambient heat, allowing it to maintain low temperatures without relying on external power sources. This feature not only improves qubit stability, but also simplifies maintenance. The cooling mechanism involves transferring heat from the target qubit to a cold qubit within the refrigerator, utilizing the temperature difference between two thermal baths for effective cooling. The findings were published in the journal Nature Physics on January 9, 2025, emphasizing the autonomous capabilities of this quantum thermal machine and its potential for real-world applications. While this innovation represents a significant advancement towards practical quantum computing by enabling more reliable and error-free computations, it is still in the early stages. Challenges related to scalability and mass production remain to be addressed before widespread adoption can occur. The economic implications are noteworthy, with a projected global investment exceeding $45 billion by 2025 in quantum technology, driven by advancements in hardware, including cooling technologies. Reliable quantum computing could revolutionize multiple sectors, from environmental modeling to healthcare, enhancing productivity and creating jobs while addressing critical issues like climate change. Researchers at Oregon Health and Science University have made a groundbreaking discovery regarding the role of perivascular cells in blood vessel dysfunction linked to chronic diseases like cancer, diabetes, and fibrosis. Their study reveals that these cells are crucial in driving blood vessel leakage and distortion, which in turn exacerbates inflammation and worsens disease outcomes. Led by Louise Bertassoni, DDS, PhD, the research, published in Science Advances, indicates that perivascular cells can sense changes in nearby tissues and send signals that disrupt blood vessel function, contributing to disease progression. To explore this, the team utilized a blood vessel on a chip model, developed in collaboration with Boston University and the Weiss Institute at Harvard, to simulate conditions such as tissue stiffening and scarring. The findings point towards the potential for early detection and intervention in the changes occurring in perivascular cells, which could prevent precancerous lesions from advancing to full-blown cancer. Bertassoni emphasized that addressing these changes early could transform cancer prevention and treatment strategies. This research enhances our understanding of the interplay between the extracellular matrix, blood vessel function, and disease progression, positioning perivascular cells as promising therapeutic targets. Historically, endothelial cells were thought to be the primary contributors to vascular disease, but this study shifts the focus to the critical role of perivascular cells as important sentinels in vascular responses. The study also demonstrated that the removal of perivascular cells led to blood vessels failing to respond to changes in surrounding tissues, highlighting their essential role in maintaining vascular health. Bertassoni's team is recognized for their previous work in 3D printing blood vessels and continues to develop realistic blood vessel models for disease study. Overall, these findings may pave the way for new treatment approaches for chronic diseases, emphasizing the importance of targeting perivascular cells. A recent study published in MBio reveals a critical mechanism by which the Zika virus enhances its reproduction. The research identifies that Zika exploits a host protein known as ANKL2, which plays a vital role in brain development. This interaction between Zika's NS4A protein and ANKL2 not only boosts the virus's replication efficiency, but also helps it evade detection by the immune system. In cells infected with Zika, ANKL2 forms clusters around the endoplasmic reticulum, creating pockets that serve as virus factories, thereby facilitating improved replication. Research led by Adam Fishburne indicates that knocking out the ANKL2 gene in human cells significantly reduces Zika virus replication, highlighting its importance in the virus's life cycle. The Zika virus's ability to cross the placenta is a key factor in its association with microcephaly, a serious birth defect characterized by abnormal brain development and smaller than expected head size. Priya Shah, an associate professor at the University of California, Davis, pointed out the unfortunate timing of Zika's presence during fetal development, which poses significant risks during pregnancy. The study also notes that NS4A proteins from other related viruses, such as dengue and yellow fever, similarly interact with ANCL2. This suggests a common strategy among these viruses to utilize host cell proteins for reproduction. Zika strategically hides in pockets formed by ANCL2, complicating the immune system's efforts to combat the virus. Additionally, collaboration with researchers from the University of Nevada, Reno, indicates that Zika also utilizes ANCL2 in mosquito cells, underscoring its significance in both human and insect hosts. The research received support from the National Institutes of Health and the W.M. Keck Foundation, with contributions from multiple institutions. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.